as you guys read chapter 12, read 12, it talks about mitosis and cell cycle. Most of your cells look like this right now. Right? Most of your cells are just doing their normal functions, making ATP, proteins, enough to survive, whatever the tissue-specific work is. Which means your stats right now shouldn't be working for the most part. Your CDK should be inactive. Okay? Your calcium are obviously being pumped because those you do need. Right? Caspases are all stuck in the mitochondria. This is the normal state. Now, a couple more things I want to start adding a little bit to this. And again, especially this PRB. We haven't talked about PRB yet. And I mentioned that some of you, they're phosphorylated and they're hanging out in the cytoplasm. Sometimes they will get, I'm going to add this guy here. Sometimes they will bump into one of the, like SOX or one of the enzymes that dephosphorylate, kinases, for example. And some of them will bump into PRB and knock off the phosphate, which actually makes it active. In a quiescent state, it doesn't matter. Like now, you're going to see that in just a second of why. So meaning if we like knock that P and activate PRB accidentally, if the whole cell is in quiescent state, there's no substrate for it. So it actually doesn't matter whether it's active or not. It's just kind of floating there. We're okay with that. So let's activate these pathways again. Okay, and see if we can follow it. We'll speed this up a little bit now. All right, so we have ligands. Now these, again, these could be cytokines sent from immune system cells, T cells, white blood cells. They could be hormones, estrogen, testosterone, progesterone. They could be growth factors. And these would be sent from different glands, pituitaries, who are also working on their own feedback. How do they know when to send? They have their own hormonal loops and things that are happening. Right? So they are gut signal. Send out a bunch of ligands. They're going to attach. Right? And again, the phosphates are just floating. Right? They're negative. So coming from ATP. Right? So there's, as soon as this happens, we have an enzymatic reaction. These get activated, shift. JAKs now activate each other. Right? And we're starting a signal. Let's just work like this one. Okay, so here we go. These guys change shape. Now these guys are going to bump into them. Again, this is just a lot of diffusion, a lot of bumping. There is no like draw. They're not all of a sudden drawn to it. They just have to bump into it. Okay? So then this kinase is going to get activated, right? And then hopefully move and activate a bunch of kinases that are sitting on a scaffold. Right? So we get transduction of signal, lots of cascade, domino effect. And then some of these kinases, hopefully, sooner rather than later, bump into a stat. Right? So, or a transcription factor. We're going to change the shape of the stat. Transcription factor. These guys, right? And again, they come together and form, let's put S in that, okay? All right, form a dimer. So far, so good, right? So then these guys are going to migrate. And they have now the sequence you need to enter. So this guy cannot, even though he's just hanging there, right? So we have activated stats. They enter in here, and they're going to start transcribing, making, or turning off genes. Okay, so what are we going to make here? We're going to make some SOX, right? We're going to be a transcription factor for SOX. We're going to also be transcription factor for cyclin, right? And we did that yesterday. So that means we're making some mRNAs. Blue. Right, so this again, hopefully a quick review. mRNAs of these two, they're going to exit as mRNAs, be met with ribosomes that are hanging outside the nucleus, right? Go through a transcription translation process, make proteins, and what are we? Oh, we're making this directly. I think everything. Oh no, that was it. Oh, sorry. Yep. Keep them close. Okay, so cyclin was a little triangle, right? And then SOX was little circles, I think. Right? SOX. There we go. So we make these two guys here, right? And they're going to diffuse. Cyclins are going to attach and activate CDKs who are just floating inactive. Right? So here's that cyclin. And socks are going to go float over this way and start cleaving off the phosphate, thus deactivating the pathway so it doesn't keep going. Good with that. So negative feedback, socks goes, okay, that's plenty. Let's start shutting you down, right? And in the meantime, we're making enough cyclins to start activating CDKs. That's where we left off yesterday. Okay, and then let's go back over here. And this could be happening at the same time or not, right? They could be happening sequentially, one first, then the other. So over here, we're also going to receive a ligand, cytokine growth factor, that changes the shape, right, of this guy. He's twisting whatever he's doing, okay? And then that forces RAS to shift. So right now you can see RAS GDP. So we're going to flip it back around, right? We're going to go this way and activate it. So it's going to be GTP, again, cousin to ATP, so guanine triphosphate, right? We'll go this way. So this gets activated. Let's go start activated. Changes RAS. RAS bumps GDP. GDP. GAPs cannot, right? GAPs cannot attach to a GTP-bound G protein, right? Good. So it just changes enough shape that this gap loses affinity, right? For the GDP-bound one, it doesn't interact with it. Because the gaps deactivate RAS. So until it has a GTP, it doesn't touch it, right? So we now activate RAS. RAS now is going to go activate whatever, right? It's an enzyme or some other factor on the membrane, releasing that second messenger, right? And again, I have this guy here nearby, so maybe it's going to attach to a kinase, changing its shape. It can activate an enzyme, or right, we had it moving, this guy moving over here, just like on that animation, opening the gated channel, releasing a bunch of calcium. Right, they rush back out down the diffusion, down the gradient, right, because over here we're pumping them in forcefully. So calcium are now able to spread around, do some things, and eventually we'll need them in mitochondria, which we'll talk about later. Okay, so this pathway is now active. Right? And this kinase could also then activate a transcription factor. See, so far, like in the middle of our board, right, that's transduction of signal, meaning reception is over here, we receive the signal, do something. We transduce and say, okay, let's everybody get up, let's work, but we need to get over here. The response only happens when there's a change. So releasing calcium is a response. Making new protein is a response, right? We can just transduce the signal and nothing happens. So back, as RAS keeps going, eventually, again, we did this also, right? We have a transcription factor, whether it's over here or just a new, maybe this guy made it, right? Again, we're simplifying. But it will go in here, and we're going to make E2F, right? We did that yesterday as well. So E2F was E squared, right? So we were going to make mRNA for E2F. That's from the RAS pathway, right? And again, the mRNA comes back out. And here's E2F protein that's made by the ribosome. We're good with that. Now I'm going to add new stuff. So this is all from yesterday. Good? So far, because the signal, the cell was quiet. The cell was like, I'm living, I'm good, I'm happy. So we're growing. Right? All of a sudden, like, okay, we need you to start moving. Like, get out of your comfy zone. Start cranking up things you need because you're going to divide. We need you to divide. We need you to repair tissue or, just get bigger. Right? We need more cells to fill in the space. So for this cells to divide, it needs stuff. Think about what it needs to divide. It needs to replicate its DNA. Right? Because synthesis phase. You have to double it before you split the cell. Which means normally you don't have any enzymes that do that because you never replicate your DNA. Each cell, under your normal condition in your lifetime, will divide about 40 times, okay, in your whole lifetime. So which means most of the time they are quiescent, they're not dividing. So that means you don't carry enzymes for DNA synthesis. Why would you? You don't need it. Right? So this is what we're cranking out. We're getting everybody ready to make all the stuff you normally don't have. So that's when you got this signal, okay? So far, so good. So here's our A2F. Now we're going to do something with this. The cyclins are now ready and they're active. So what do they need to do, okay? Here's E2F is a substrate for PRB, okay? But, okay, the only PRB that this can attach to is the active one, okay? So what happens is, if there's an active PRB, which means it's dephosphorylated, it will attach to E2F, like here. Right? You'll find E2F, and it will attach to it, like that. Why is that good or bad, or what's that important? When it attaches, it holds on to it. E2F is a transcription factor that is needed to make other genes that are necessary for synthesis and mitosis and other things. So if you're holding on to E2F, it can get back into the nucleus, which is where we need it. Right? You with me so far? So how do we prevent this from happening? These guys. One of the main things, not the only one, but one of the main things of an activated CDK is to phosphorylate and inactivate PRB, right here. So here's a CDK with a cyclin that's going to attach, phosphorylate, and inactivate because we don't want it holding on to the E2F. Because this cell got a signal to divide, so we want it to divide. So we don't want this to happen, which is why we made these guys first. Because when these guys exist, they're going to go float, attach, and make sure all PRBs are turned off so that they don't grab that E2F. All right, so we're going to do that. We're going to add a phosphate to this guy. Boom. So now he's not going to grab the E2F. Yes? And then E2F can get back in the nucleus and make a bunch of genes that we do need, and the cycle progresses. So until PRB is inactivated, until CDKs are activated, until E2F is made, there's a checkpoint. Right? So when you read my paper and you read chapter 12, there's a couple of checkpoints in your cycle. Let me just get to this point real quick. Right? Remember, it's like a pie. They're not like the right sizes, but just to get an idea. Right, Rossi, like that, telekinesis. So the cycle, most cells from here exit out and go to G0. That's quiescent. Right? Most cells are over here. When they are signaled, they go back to this. So G1 is where all of this is happening. Okay? And then right here, there's a big checkpoint. This is where we want to make sure, okay, do we have all the E2Fs made? Do we have all the PRBs turned off? Do we have all the cyclins active? Like, is everybody ready? And until everybody's signal's ready, this will not continue into the S phase. Right? So it's sort of damage control. Okay? Because for some reason, if PRB is active and is grabbing E2F, cycle stops. Which we'll talk about in just a second of what happens when it goes wrong. Everybody okay so far? Yes.
So the CDK attaches to the PRB so that the E2F cannot so that
join.
they keep making E2F.
Correct. Good. Yes. So what we want this phosphorylated so that it cannot attach to the E2F, right? Because E2F is needed to make, right? E2F, and I don't have it on the board here, but E2F actual protein will actually act as a transcription factor for a bunch of other genes that we do need for the S phase. So the E2F transcription factor is present we don't have enough staff, enough people, enough guys, enough workers to actually produce and do synthesis of DNA, right? So there's no reason to continue the cycle because E2F isn't making the right guy. Is that good? Everybody good? Right, so we want cyclins to phosphorylate or PRBs so they're staying shut. Okay? E2F is free to go, makes a bunch of stuff, we're good, cycle progresses, everybody is good. But now, right, when we want to shut it off. So this is good. If everything is good that E2F makes it, we continue to S phase. There's another checkpoint right here. Right, so a checkpoint. This checkpoint is kind of similar to this, another pathway. Make sure we have everything we need for the M mitosis part, right? And if we have everything we need for that, mitosis happens, cell divides, and then we need to shut all this off. Okay? So how do we shut it off under a normal situation? We have to stop getting signals from the pituitary and the hypothalamus, right? Which means these guys get shut off. So we're going to shut this guy off and shut this guy off. Right? But now again, these kinases are all activated. So hopefully there's enough SOCs at this point and other proteins that are dephosphorylated everybody by now. Right? Signal is done. So we're going to dephosphorylate everybody so that this stops happening, this stops happening. We stop making cyclin, stop making SOCs, shutting this process down, which means these guys are no longer attaching to CDKs, like we're not making any more cyclins, which means they dissociate from PRB, and PRB gets dephosphorylated. They're now active, grabbing all available E2Fs that are floating. Boom. E2F, E2F. So which means, right, they're going to go grab these guys, get them out of the cytoplasm, so they're holding on to it. So E2F cannot enter back to here, and cycle stops. Right? Now these guys over here, because E2F stops making the right stuff, so we're back to where we ended up. Now we got to shut this guy down. So here's, right, look where we are. So we shut down the receptor. Does that shut down RAS? No. And as long as RAS is active, it's going to keep hydrolyzing, so that guy is going to keep releasing, that calcium is going to be made, this transcription factor E2F is going to keep being made. Do you see that? So what happens if PRBs are holding on to the E2F, but RAS is still active, so we're still making a bunch of E2Fs from here. Do you guys see what I'm saying? Because this transcription factor is still going. So everybody else is doing the job, but we keep making more and more E2F. If that happens, right, we never turn RAS off, and we keep making E2F, which means E2F keeps running back in and making all the stuff that we need for these phases, what's going to happen? Cell keeps dividing. So you might say, well, if there's no signals anymore, why is it doing that? Because RAS is still active. Right? Now, why is that? And what happens then? What happens if this keeps going? We leave it like this. You're going to keep dividing. Now you have a transformation happening. Transformation is when a healthy cell transforms into unhealthy. Right? And now this cell is going to continue dividing, even though the parts are okay, but this is the problem. RAS pathway is now damaged. It's always on, and now we have a body mass and tumor forming. Right? So how do we shut it down? We have to shut it down with RAS. So one of the current most leading research, like Fox Chase and other big places who do cancer research, they're trying to figure out how to shut down RAS. RAS is causing a lot of cancers. I don't want to give you numbers, I don't know, but it's a lot of cancers. Is RAS, and I'll show you what the other ones are. RAS is an oncogene. Okay, we're going to read about that today. Oncogenes are directly responsible for cancer. So this is, RAS is the single, this is why the cell is dividing, because RAS is on. So that is an oncogene. Yes.
how are there some cells that have damaged RAS and some that don't? Is it like
Okay, we'll talk about
hereditary?
how RAS can be damaged. Yes, okay, we're going to talk about that in a second. So let me just say it. Now, on a normal situation, how do we shut RAS down? So we have to be able to shut it down, right? Under normal situation, GAGs have an affinity for RAS GTP. So when it's active, GAP can actually attach to it, right? Under normal, it has an attachment site, and it will cleave this thing backwards, right? It will go this way, and that way it changes the RAS shape back to where it was, so it loses the affinity for GTP. GTP gets released, GDP comes in, sits there, shuts this off, which means now it can no longer work with that, so when we stop making this, now we shut that thing down. So how do we damage RAS? Okay, so the most extreme case is that you, as a baby, got a mutated RAS gene over here. And your mutation is in every cell because you got it from your parents. You will not survive. That's fatal. I mean, the baby will never be made because RAS will be always on, so it'll just die, right? That's the extreme. Now, what if you have a partially mutated RAS? Because that's what happens. Sometimes you get some mutations as a baby, but not enough to make a mutagenic RAS. What happens then? Throughout your life, as you exposed to other things, some of your cells can get mutated. And then you hear this enough, radiation, radicals, the diet, your whatever, accident, right? By some chance, by some whatever, and there's a million reasons why some people get cancer and others don't, right? Some of your cells can mutate because they're just more radically damaged. They're the ones that are always exposed to the same area of blah, 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 right? That in your liver or in your lung or in your whatever tissue, those cells are getting bombarded by damaging something, and their mutation, RAS, maybe you already had a couple of 5 mutations, and now you got one more, and that was enough to now mutate your RAS gene, which mutates your RAS protein, and let's say you lost the binding for GAP. Let's say the mutation was in this region. Now those cells haven't actively turned on RAS, and that's where you're going to form a tumor.
And what kind of like a disease would that look like?
Cancer.
Just always, forever and always.
Yes.
But that wasn't turned on. That RAS wasn't damaged at the beginning of the life.
Partially damaged.
Okay.
Correct. Partially damaged. Any single mutation will not cause cancer. You would need a series of them. Mm-hmm. So when a cell first transforms, does it automatically mean it goes to cancer? No. Why not? Because there's a lot of checkpoints. A cell has many ways to prevent itself from being cancerous. And that's where tumor suppressor gene comes in, which is the second half of what I'm talking about and what's in my paper, right? Because you're right, otherwise, oh wow, mutated RAS cancer. No, we do have a lot of controls before that happens. Like most people, like in our lifetime, most people will develop transformed cells. Most people, like 90%. But most of those people will not be cancerous because this will get cleaned up, right? So I would say out of all the transformed cells, and I took that cancer biology class, that's where that paper is, the statistic was like literally like 0.0001 or 0.001% of transformed cells who actually become cancerous. Like it's actually really, 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 really small percent because it takes a lot of steps to become a cancer tumor. Does that make sense? But to be a transformed cell doesn't take as much, but we can control. Right, good question. Yeah. Is everybody okay so far? Yeah, well, I'll show you the controls. Yeah.
The JAK stat pathway.
Yeah.
It ends up making cyclin, which goes.
Again, it makes a bunch of other genes.
Yeah.
I'm just
So,
throwing an example.
The beginning of JAK and the beginning of the RAS one, they have nothing to do with each other.
They crosstalk. So that means some of these might, like maybe this kinase gets activated by RAS, but then it will actually phosphorylate that kinase that talks to JAK's part pathway. That's what crosstalk means. So maybe this kinase got phosphorylated by this pathway, which still will lead to the same cascade, even though the JAK stats weren't actually the one activated. That's what crosstalk means, that one pathway can influence the other. And RAS pathway does influence a bunch of other pathways, Because the question is, why has it been so hard to find cancer treatment? Because that's why. First of all, RAS is in every cell of your body. So how do you target that RAS? Right? Because it'll attack all your healthy cells. If you put a drug in to kill RAS, it's going to kill every RAS in your body. Do you know what I mean? So it's very difficult to find a drug. And then you shut off this RAS, but it interacts with like 10 other pathways. Right? So now you might have fixed this problem, but now you created like 8 other problems. Right? And that's why it's so hard to treat this, because there's just so much going on that you, right?
What makes the ligands detach from the transmembrane proteins?
Okay, so they come and go. Concentration is the big thing. These guys detach and attach at all times. You need a threshold. There has to be enough density for these to keep firing. And a lot of these have a half-life. They just die of indigestion. Okay. Yeah. Yeah, good question.